Your Majesty, Excellencies, Minister, other dignitaries, ladies and gentlemen, it is my pleasure to have the opportunity to deliver this talk to you today. Rector Bjarklev asked me to talk about a topic of general interest, something that had some scientific and technical content, something related to the DTU, and something that was interesting and might create some dinner conversation. So I chose this topic. Now, 40 years ago, when I was being raised in Iceland, I was taught about the Vikings sailing their ships around the North Atlantic. And when Icelanders sailed to visit royal Scandinavian courts, they would bring gifts, which might be a book written on a cowhide or a poem to please royalty. I chose one such poem to recite here to Her Majesty from Hávamál, which is Odin's speech or Odin's advice, which is a set of poems that has common sense advice to people. I'm going to read it out in Icelandic, maybe Old Icelandic as well as I can, to simulate what her predecessors might have heard some centuries ago. For the rest of you, I'll put the translation up in English as I read it out. So here we go. Deyr fjö, deyja frændur, deyr sjálfur hið sama, en orðstír deyr aldrei, hveim sér góðan getur. So this is classical wisdom about the fact that the reputation you generate will supersede you, and it better be a good one. And as you saw, with all the honorees here today, this is what professors strive for. So this is a poem that is probably 12 to 1500 years old. Its wisdom today is just as applicable as it was 1200 or even 1500 years ago when it was written or composed. And this leads me to another proverb, Icelandic proverb, Góð vísa aldrei of oft kveðin, that translates into a good poem can never be recited too often. Fast forward 1200 years, Icelanders still travel to Denmark to speak to royalty. We use a different means of transportation, and we may travel from further afar, and our messages may be written in a different medium. We now write knowledge in the form of DNA. Here is the code to the insulin gene written out as a poem and called Ode to Insulin. It might be called the Ode to Joy because it creates 3 or 4% of Denmark's GDP. That, therefore, leads to a modern Danish proverb that says, a good gene can never be expressed too often. Especially that for insulin by yeast, if you're with Novo, or especially that for cellulose in filamentous fungi, if you're with Novozymes, or especially those that make ethanol in yeast, if you're with Carlsberg. So therefore, a lot of wisdom, at least knowledge, is written in the form of DNA, and it carries future and fortune. In fact, it's a little more even complicated than that. Our entire life, in some aspects, is coded in our DNA. And this is a quote to Craig Venter, who led the public effort for sequencing the human genome in 2000, where he says, life is a program written in DNA. Now, therefore, DNA reveals the past and predicts the future. And as our prorector said, Prorector Wegener, get used to it, learn how to read it, and use it practically. Let me give you some examples. The majority of the Icelandic nation has been genotyped, and therefore its entire family tree, its pedigree, has been mapped out. And interestingly enough, this pedigree is 99.5% accurate, which apparently cannot be said about some other European nations. We can now go into museums, drill holes in the skulls of Neanderthals, isolate the DNA, sequence it, and place the Neanderthal in its proper place in the phylogenetic tree of high-end primates. We can now sequence infectious agents like Salmonella or Staphylococcus aureus around the world, find the mutations, see how it spreads around, and map it out. And in fact, the DTU is involved in a project called Google Maps Bacteria. And this is done by tracing the DNA. And finally, DNA itself has a genealogy. We know that we inherit from our father and mother the nuclear DNA in roughly even proportions. But the DNA in the mitochondria is only maternally inherited and only comes from the mom. And therefore, we can trace back our maternal background. The DNA sequencing in Iceland reveals that almost half the mitochondria in the Icelandic population are Celtic in origin. On the other hand, the Y chromosome that comes from the father, the male chromosome, is over 90% Nordic. Therefore, DNA reveals the past and predicts the future. Predicting the future, of course, means you have to understand some of the basic properties of DNA. I don't have to introduce Niels Bohr to Danes and the fundamental work he did for chemistry. It turns out that exactly 100 years ago, in 1913, he started working on the model for the atom, and it took, I think, 10, 15 years to perfect it. And we're all familiar with this, with the neutrons and the protons in the nucleus and the electrons circling around it. And this conveys fundamental information that had led to much progress in chemistry and its use. 100 years later, can we do the same thing? Can we make predictive models, conceptual in the beginning, and then mathematical, that are useful for practical purposes? It turns out that we are in the process of trying to do that. And this seems to be a three-step process where we take a living cell, we rip it apart, isolate all its components, and we find out the system that underlies its function. And Professor Brunak mentioned some of that in his taped remarks. We then try to convert this into a mathematical format to compute cellular functions. And since this is a technical university, we'll show some mathematics. When you don't know much about the cell, everything is possible. It can be anywhere in that three-dimensional space depicted. But when you have reconstructed all the functions of the cell, you figure out that the cell cannot just do anything. It's restricted in its behavior, and therefore computing its behavior is confined to a space that we call a solution space, as shown in that blue object in the middle. And then we use optimization procedures to try to predict the future behavior of an organism. In some vague sense, similar to the atomic model of Niels Bohr, where the orbitals are probabilistic in their nature. Now, this field is only about 15 years old, and I'm going to show you its progress curve as measured by the number of publications in the field that have predicted or interpreted data. There was some pre-genome era activity on mathematics and small-scale networks. In 2000, the first genome scale model came out, and we were obsessed with algorithms and computation. Then reality set in, and between 2005 and 2010, these models were validated against real data. And somewhere around 2010, 3 years ago, this field got to the stage of development where it was predicting real biological phenomena. And therefore, we have the engineering dictum that nothing is as practical as good theory, because you just calculate, you design, you don't always have to prototype and experiment. And the Center for Sustainability, that I'll tell you more about later, was started about 3 years ago when this became possible. In terms of dinner conversation, you may notice the doubling time of this field, which is a knowledge-generating field, is 3 years, and compare that to the speed of improvement in computation or computer performance, which doubles every 18 months, and sequence information even faster than that. What opportunities does this create? It is undoubtedly going to impact infectious disease and how we deal with it over the next 10, 15 years or so. It's going to impact our understanding of human metabolism, its involvement in health and disease, and under stressful conditions, such as during sports, being in space, or studying for your finals. The CFB itself is focused on engineering into microorganisms metabolic properties that allow us to produce various compounds, as I'll show you in a moment. So the near-term opportunity for the practical use of this capability and this information is in the chemical industry, and try to generate a renewable chemical industry. We are all aware of the need to develop sustainable lifestyles. We associate big factories with this industry, industrial chemicals, but we also have fine chemicals, flavors and fragrances, and we even have pharmaceutically active molecules. So there's a big spectrum of molecules that can be produced biologically. The structure of the chemical industry that we are becoming familiar with is depicted here. The oil that comes out of the ground is mostly burned. 90% of it goes into fuels. 10% of it goes into the chemical production. And that is actually a gigantic industry, I should say. And most of us are not aware of the fact that it's over a $3 trillion industry a year and literally represents 5% of the global GDP on this planet. The products in this sector are over 3.5 euros a kilo, and they can now be economically produced in many cases biologically, and therefore provide an economic alternative to chemical production from petroleum. This possibility led to the Novo Nordisk Foundation funding a center here at the DTU called the Center for Biosustainability. And that happened in December of 2010, when Birgitta Nauntofte, pictured here, with an envelope with a 700 million Danish kroner check in it, handed over to the previous rector, Lars Palsson, who quickly proclaimed that as the biggest Christmas present he had ever received. Now, the vision that is being put forth here is that microbial factories will change the world. They will affect how we live in better harmony with our environment. So I'll talk a little bit more about the idea behind this center and the plan that underlies its operation. The overall goals of the center are actually very, very simple. First is to determine how many of these molecules in the chemical industry can be produced biologically. So if you're a businessman, you would say, what fraction of that market is addressable? If you're an engineer, you're more interested in shortening the design and the developmental process that leads to a microbial factory. And I'll say more about that in a moment. We are using 3 cell types to make things: Escherichia coli, yeast, and Chinese hamster ovary cells that are used to make therapeutic protein. And I don't have to remind any Dane that yeast is a natural cell factory for beer. We'd like it to be able to produce more things than beer. Now, a little bit technical material. In the blue boxes in the middle, we have the workflow by which the center operates. First, we have to identify our target molecule. Then we have to find the biochemical pathway that makes it. Then we have to construct the cell factory. Then we have to optimize it, and if it works, it goes into scale-up. As you can see to the sides, there's a lot of experimental and computational technology that has to be developed to support this workflow. The workflow is not exactly linear. It is an iterative workflow, as I'll describe on the next slide. But what is important about it is the fact that today it costs $50 million and takes 5 years to build a cell factory. Our goal is to bring that down to 2 years and $20 million. And anyone in that industry knows that if that is achieved, we will fundamentally transform it and maybe enable that vision to be realized. Just briefly for those of you more technically oriented in the audience, this iterative loop is comprised of computation, these genome scale models I mentioned earlier, genome editing, where we systematically change the DNA, and then we characterize the cellular function that results from that editing, and then we do genome scale measurement to see how that cell behaved, then we model that data, and around we go. So this is a very interesting integration of a diverse set of scientific disciplines and technical or technological skills. Now, we got funded just before Christmas 2010, so we started implementing the center in 2011. Let me just show you briefly what has happened since then. First, we got the check. Then, we built the center very quickly up in Hersholm, and we opened it short 10 months later. And as you can see in that picture, if you have good eyes, there is Anders Bjarklev, had become rector at that point in time. 15 months after we started, the CFP existed, and we had hired the 60 people or so we were funded to hire. Fortunately, the foundation was impressed by our progress and parted with some more money for us. They gave us over 400 more million Danish kroner that allowed us to expand our activities, to look at Chinese hamster ovary cells, and build an international network of collaborators. We have just recently, 3 months ago or so, moved the expanded version of the center into a new building, also up in Hersholm. So this has been a rapid process and a very pleasant one so far. This shows the faces of the people that comprise the center, and a little diagram in the middle that shows its growth. And if you have good eyes again, you look at the third bar in there for 2013. At the end of this year, we should be about 170. And that's the level of funding by the foundation. And after that, we should grow based on external funding. And somewhere in 2016 to 2017, the center will move back to campus into a new building that is in its early design stages. I have to apologize to those that thought they had a good position in this composite picture by overlaying them with that diagram there. Let me just talk a little bit about this center, why it is unique, and why it is in a position to take a worldwide leadership in this field, and hopefully do great pride to the DTU community, and be useful to Danish society largely. We have very strong driver behind us. Biosustainability is a strong political and socioeconomic driver. The foundation gave us a 10-year time frame and a big financial muscle, over 1 billion Danish kroner to do this. We've had unstinting support from the DTU Rectorate in every possible way that has removed many obstacles that otherwise would have been in our way. We've been able to attract first-in-class scientific directors and very quickly establish a world-class team of professors. And we are fortunate by the fact that this is a relatively new field, and the academic leadership in it worldwide is narrow, and most of it is now associated with the center. Again, if you were a businessman, you would describe that maybe as M&A activity. We are truly one in a kind in the world at the moment. But the $3 trillion market is not going to be lost long on others, and there will be competition, and therefore we are trying to work as hard as possible and as fast as possible to build this center. It has an unusual organization in that it is a hybrid operation where roughly half of it is focused on academic research, PhD training, publication, and half of it is focused on translational research, where we hope to do or lead to good things and commercial activities off campus. This is also in tune with the times. As you know, the world is in a challenged economic state, and most countries are now looking to their universities for innovation, source of new industries, source of new activity, and we hope certainly to play a part in that role. Briefly, give you a sample of the sorts of products we are after. We are either after, so on your left, high volume products that are cheap, like 3HP. This goes into acrylic acids and makes disposable diapers, for instance. It makes the screen on your cell phone and other structural materials. If you're lucky enough to have a swimming pool, you might cover it with some acrylics by 3HP. Resveratrol is more of a fine chemical made by a company here in Denmark called Fluxome. This is the active, or believed to be the healthy ingredient in red wine. Serine is an amino acid that goes into cosmetic products. It makes us look beautiful. And then IgGs, which are protein that are therapeutic. So we have a spectrum of products that we are focusing on. Finally, I was asked to make some observations about Denmark, as I've been operating here for 3 years now. I should say my parents met in Copenhagen, and I was raised in a home where I learned a lot of Danish values, and I actually learned Danish in school also when I was being raised, so I didn't come here as a total stranger. I knew a little bit about Denmark, but I made some observation as an adult of Denmark since I came here, and this, of course, is in context of what I'm trying to build here. First, Denmark, and not everybody realizes this, but Denmark is possibly the most experienced country in the world with industrial biotechnology. This means that we have a depth of workforce and culture here related to that. Most Danes at some point in time have had exposure or worked at Novo Nordisk, Novozymes, Danisco, Christian Hansen, the beer companies, and so forth. Danes and Scandinavians in general tend to have a practical outlook, so we are not ashamed to practice translational medicine in this center. One thing that really did surprise me was to find out how big of a role private foundations and companies play, how big of a role they play in public research and development. This is much more than what I have experienced in the United States, as an example. Yes, Danes have strong vision. They build industries like windmills. They go into China and India early into these emerging markets and build dominant positions there, from which Denmark now benefits. And I think we should all recognize that biotechnology is a large fraction of the GDP here, and I think Novo Nordisk alone as a company is something like 4% of Denmark's GDP. So the bottom line here is that life sciences will be a significant part of the future socioeconomic development as we approach sustainable lifestyles. And Denmark is in a good position to take a leadership position here, and we all hope that the CFB plays an important part in that development. Now back to the beginning of my talk as I close it. Here are 3 Danish Viking ships full of happy Danes sailing into the sunset, looking for future fortune and future, reciting poems to each other that are loaded with classical wisdoms. And then as they move into the future for 1,200 years, well, oops, it wasn't the sun that they were finding. Maybe it was a nucleus filled with DNA that is full of valuable messages that we can now exploit. And therefore, we have this proverb, a good gene can never be expressed too often. Thank you very much for your attention.